Okay, so once again, I'm going to use this to highlight the importance of giving an introduction, then your main content, and then your context or conclusion. So, if I'm talking, or if you're being asked to discuss how the adaptive system achieves antibody diversity, or it may say something like, how does the B cell achieve high diverse antibody repertoire, something like that. It's the same thing. How are we able to create or develop enough B cells to deal with the wide range of pathogens that exist in the environment? So one thing when you're addressing this issue is you could talk about the fact, look, the innate response is not suffice to eradicate any infection. We need the adaptive response. So in order to eradicate an infectious agent in a specific way, we need the adaptive response, which includes B cells and T cells. And just to highlight that each B cell and T cell has its own unique B or T cell receptor. Okay? And it does that through a process of somatic recombination and junctional diversity. So you have to remember, it's just not about answering a question. You're given the opportunity to convey your understanding, and in this case, what you're trying to tell the examiner is, I know that each B and T cell has its own unique B or T cell receptor. And I know that it didn't start out like this. The way that we're able to achieve antibody diversity is the fact that each B cell goes through a process of somatic recombination alongside junctional diversity to help each B cell, B or T cell, to obtain a unique version of that gene and therefore a unique receptor. Okay? Now, in terms of this essay, I want you to give a context, okay? So the whole rationale to create antibody diversity is there are limitless amount of pathogens that exist in the environment, okay? Now the problem is, if we had one gene for every pathogen that we're exposed to, you would have an insane amount of genes to try and deal with that, okay? You just wouldn't be able to fit it in a cell. So the way the body has evolved to deal with this is it uses one gene, but it edits that gene in every B or T cell in your body. Okay? So considering the diverse range of antigens that we're exposed to, the concept that we'd have one gene to respond against one pathogen, it's not possible. So the way the body overcomes this is through the process of somatic recombination of the variable region of the B cell receptor, okay? Alongside junctional diversity, which we'll cover. And by doing this process of somatic recombination and junctional diversity, we're able to achieve a vast amount of different types of B cells with different types of B cell receptors. And we're talking up to 10 by 10 to 11 or greater, okay? Now there's one slide that I need you to know during this process to answer this question, and we'll cover that. Now, just to kick off the main content of the essay, what you need to appreciate is that the antibody or immunoglobulin has two specific genes generally that are involved in. One is the heavy chain. The other is the light chain. Okay? So here you see the heavy chain of antibody, and here you see the light chain. And what happens is them two come together to form immunoglobulin. Now, in terms of those two genes, I need you to appreciate this, okay? For the heavy chain, everybody has two copies. One from your mother and one from your father, like any gene. Okay? Now, for the light chain, we have two types of light chain gene. One is kappa and one is lambda. But with kappa and lambda, we also have two copies of each gene in our bodies. Okay? So here you see we have a copy of the heavy chain from our father, a copy from our mother. For the light kappa chain, we have one from our mother, one from our father. For the lambda chain, we have one from our mother, one from our father. And these are just random nucleotides, okay? So that's to start us off. Now, what happens is, during B cell development, they all start with the exact same version of that gene, okay? Now, what's going to happen is, it's going to try, and each individual B cell, I must say, will do this in different ways, which is why they all have unique versions of the gene. But the way it starts is, it will take one copy of the gene, okay? So it'll either start with your mother's copy or your father's copy. In this case, we'll start with the father's copy, okay? Take you through that. Okay? So, it starts with the father's copy. And the way it does it is it uses enzymes called RAG1 and RAG2, which are symbolized by the scissors. Okay? So RAG1 and RAG2 are recruited, and this is a completely random process, folks. RAG1 and RAG2 will choose to cut the gene in a specific way. But it always starts in this process. The regions of the heavy chain, it selects two random regions, one from the D region and one from the J region. Okay? And it cuts out that piece of DNA. Okay? That's now out of that, in this particular cell, the part of that gene is gone forever. Okay? No longer in the cell. And then what happens is random nucleotides will either be removed from the sticky edges of the D or J region, or they'll be added. In this case, there's some added. And they then come together. And the addition or removal of nucleotides is called junctional diversity. And in some cases, it'll be referred to as somatic hypermutation. Okay? So, that process is D to J recombination. The next step is V to DJ. And again, RAG1 and RAG2 are involved here. And they will cut between the D region and the selected V region. And that's cut out, and that is then lost forever. Okay? Again, nucleotides are put in or taken away. And now we have complete recombination. Okay? So for this particular B cell, that's the gene it'll have for the remainder of its life. Okay? Now we need to transcribe it to a protein. Okay, so you put it into messenger RNA, you have gene splicing, and in the end you have your heavy chain. But the one thing that needs to get checked at this point is, does your heavy chain actually work? So, the heavy chain gets attached to a protein called the surrogate light chain. So the surrogate light chain gets attached, we then express it on the surface of the cell and see, does it work? If it works, then all is good with the world and we can stop the process. Okay? So, if that works, great. That's the process finished. We only need one copy of this gene. So, if the father's recombination of the heavy chain worked, then we turn off the mother's copy. And that is called allelic exclusion. Okay? So, that's it. The heavy chain rearrangement is complete. But, okay, it's rearranged. If it's successful, then the cell will proceed to light chain rearrangement. Okay? That'll be heavy chain finished for now. But, if the gene rearrangement didn't work, the father's copy gets switched off because it didn't work. We then move towards We then move towards the other copy of the gene, the mother's copy, and we try to rearrange that. If it works, then we can progress to light chain rearrangement. If it doesn't work, then the cell has to die because we've no more copies of that gene. Okay? So that's heavy chain rearrangement. Now we're moving on to light chain rearrangement. And as I said before, we have two types, with a kappa and with a lambda. And of course, we have two copies of each of those. So we always start with the kappa light chain, and again, it's these enzymes RAG1 and RAG2 that are involved. They pick two random parts of the gene, cut them out, and they're lost forever. Again, nucleotides will be inserted, and at that point, light chain rearrangement has happened. But, again, need to check if it works. If it works, what will happen is the other three genes will be switched off, okay? Because we don't need them anymore. And once that is successful, the light chain will then interact with a heavy chain and will proceed towards negative selection, okay? At that point, the B cell has undergone successful heavy and light chain rearrangement, and now we're able to produce a functional immunoglobulin. And what that B cell will then do is go through positive selection and negative selection. Positive selection to check that it's able to respond to antigen, which we've just done. And the negative selection is to ensure that what we've just created does not respond against self-antigen. Okay? So that's what will happen. But let's say, for instance, that gene rearrangement wasn't successful. What happens then is that will get switched off because it wasn't successful. Then we move on to the mother's version of the kappa gene. It undergoes gene rearrangement, and if it's successful, the other two will get switched off. If it's not, the other two will get switched off, and then it will progress and undergo the negative selection process. The whole system will be done at that point. But if it's unsuccessful, we move on to one of the lambda genes. And if it's successful, the other version's turned off. If it's and at that point, it can then progress towards negative selection. But if it's not successful, then we're in the last chance saloon here. If it's undergo gene rearrangement and is successful, again we progress. But if it's not successful, then the cell has to die. Okay? And that's the process of immunoglobulin gene rearrangement. Okay? And this is a summary of that process that I've just covered. And again, be aware of once this process is done, we undergo positive selection to check that it works, and we undergo negative selection to make sure that it doesn't respond against self-antigen. And I explained this earlier. Once the B cell then, at that point, becomes activated, it goes through the process of gene editing to get that perfect fit. Okay? Then can undergo class switching, as I said before. Now, this is the slide you need to incorporate into your answer. Okay? Because what it conveys to me is that you understand that when you undergo V, D, and J rearrangement in the heavy chain, the amount of possibilities are 11,000. When you undergo V and J rearrangement in the light chain, the amount of possibilities are 320. And overall, if we just do V, D, and J rearrangement and B and J rearrangement, the possible amount of different types of B cells or B cell receptors that we can create is limited to 3.5 million. Now, 3.5 million is good, but it's nowhere near enough to cover the amount of antigens that we're going to encounter. So how does the body go from 3.5 million to an almost infinite number of different types of B cell receptor possibilities? It does it through junctional diversity. So that process of somatic hypermutation, where we're adding or removing nucleotides in the gaps of the VD&J and the gaps of V&J, that is what gives us the ability to create vast diversity. So that's why I need you to use this slide to convey your understanding of how we create the diversity and the importance of diversity. And just as another point, I need you in the introduction to highlight why we need diverse range of B-cell receptors. Okay, and if we were to give a context for this particular example, it would be what happens when you cannot create B cells efficiently. Okay, and of course we have B cell defects here, but we also have to appreciate that if you have a T cell defect, by default you won't be able to sufficiently activate B cells. Okay, folks, so that's gene rearrangement. That is how we create a diverse repertoire of B cell receptor genes. Now, I said I would cover, this is the last bit I'll cover, I said I would cover endogenous antigen presentation. Now, if you get a question on antigen presentation, you need to give, in the introduction, you need to give some sort of context. So be aware of how we respond to a virus. In this case, we're going to cover viral presentation. So in that introduction, I want you to lead me up to the point of presentation. I want you to say that the virus was able to overcome the physical and chemical barriers. Its PAMPs bound to pattern recognition receptors. That caused the release of interferon. Interferon mediates effects by reducing gene expression and increasing cell death of viral infected cells. And you say, and on top of that, the interferon will activate natural killer cells, and natural killer cells will kill infected cells through the mechanisms that I discussed earlier, by scanning for MHC1, and those cells that don't express MHC1 will be killed. Now, also to highlight that that the innate response is the innate response has a role, but there's also a need for a tailored response, and that dendritic cells are useful for presenting of antigen. Now, in this case, what we're going to deal with is an infected cell. So when we're dealing with an infected cell, we're dealing with presentation on MHC1. So, at the introduction, you've told me all about how the cell responds once it gets infected, release of interferon, possible role of natural killer cells. Now you're going to tell me what happens once the virus is in the cell. It's in the cell, and what happens is it will start to replicate, produce its own proteins, and when it produces its own proteins, it will then get processed through the immunoproteasome. Now, the whole function of a proteasome is to break down proteins that the cell no longer uses. Okay? It takes the proteins into the proteasome, breaks them down into amino acids, and subsequently then allows the cell to recycle the amino acids. Now, the difference with the immunoproteasome is we don't want that. We want the proteins that exist within the cell to be broken down into short fragments. If we break these viral proteins down to amino acids, the immune system won't have the ability to recognize it. So, the immunoproteasome will break down the proteins into short sequences between 8 and 12 amino acids long, so that we can isolate the epitope. That's the part of the viral protein that our immune system will recognize. Okay? So it breaks it into peptides, and the way it's able to break down, the thing that helps the immunoproteasome break it down into peptides are things called heat shock proteins. Okay? Heat shock proteins will prevent the full degradation of the protein. So now we have the epitopes. That's the part of the antigen that our immune system is going to recognize. The next thing we need to do is get it to the endoplasmic reticulum. When it gets to the endoplasmic reticulum, it enters through TAP. Okay? Now, it enters through TAP, and when it enters through TAP, it then goes through a process of getting loaded to MHC1 and then delivered to the surface of the cell where it is presented to CD8 T cells. Now, whilst the antigen was being degraded and being transported to the endoplasmic reticulum, MHC1 is beginning to be composed. Okay? But the problem with constructing MHC1 is it aggregates back on itself. MHC1 is composed of an alpha subunit and beta 2 microglobule. And here you see the alpha subunit, alpha 1, 2, and 3. And the problem, as I said, is the alpha subunit has a tendency to aggregate in itself, which makes it difficult for beta 2 microglobulin to access its binding position. So, how are we going to be able to enable beta 2 microglobulin to attach to the alpha subunit? The way we're able to do it is through things called chaperones. Okay? And the chaperones involved initially are connexin and ERP57. I mean, in basic language, what these chaperones are going to do is stop MHC aggregating by holding it up. And when it holds it up, it enables beta 2 microglobulin to slot in. Okay? And at that point, we have MHC1 functionally composed together. And once beta 2 microglobulin binds, the peptide binding groove on MHC1 becomes exposed. And we need it to become exposed so the peptide or the epitope can bind to it. Now at that point what happens is calnexin gets removed and calreticulin gets attached, and then they go through a process of interacting with taposin. And taposin is this component that stabilizes the peptide loading complex through binding DRP57. The peptide loading complex is MHC, calreticulin, ERP57, taposin, and TAP. Okay? And what happens is taposin stabilizes TAP, and the reason, what basically happens is taposin will bring TAP close together, and the reason it does that is to enable taposin to get access to the incoming epitopes. And the peptide loading complex is taposin, calreticulin, ERP57, TAP, and MHC1. And the job of taposin at that point is to assist the transfer of the incoming epitope from TAP onto taposin will facilitate the removal or the transport of the epitope from TAP onto MHC1. Okay? And once that's happened, taposin loads the epitope on the MHC1. And at that point, once the peptide binds to the peptide binding groove of MHC1, the peptide loading complex disassembles. And it disassembles in a way that it releases MHC1 with the epitope. And at that point, the MHC1 loaded with the epitope makes its way towards the Golgi apparatus. It gets packaged and transported to the surface. And when it gets transported to the surface, at that point, the MHC1 with the epitope is exposed to the outside of the cell. And the CD8 cell that's coming past will interact with the MHC1. And subsequently, if it is the antigen that the CD8 cell is designed to bind to, it will release granzyme and perforin and kill that cell. Okay? So that's the endogenous antigen presentation pathway. Now, in terms of context in an essay like this, you need to be able to tell me how certain pathogens are able to evade the immune response. Okay? I think we were asked earlier about how a worm can exist. It's because they have ways to basically hide from the immune system. And there are a lot of viruses that have very sophisticated ways to evade the immune system simply by avoiding presentation. If the virus doesn't get presented on the surface of the cell on MHC1, then the immune system will never know it's there. But essentially, what will happen is, I think it's in lecture 4, there's a slide that will show you what pathogens are able to evade which part of this process. If a pathogen, for instance, is able to inhibit the immunoproteasome, then the viral antigen never gets broken down and therefore never gets presented. If the virus is able to inhibit TAP, then the viral antigen or viral epitope is never able to be transported into the endoplasmic reticulum, therefore it never gets presented. So for that final part of your essay, I need you to talk about that. I need you to tell me, you've told me about all the innate response, the natural killer cells, all the above in terms of an introduction. Then you have told me the main content in terms of what I just talked about here. You know, the immunoproteasome, the MHC1 assembly, the presentation of MHC1 on the surface. And then for your final conclusion and context, I need you to tell me what pathogens inhibit what part of this process and how that can impact on the immune response to a virus. Okay, folks? For me, that's all I'll cover today. I'm happy to answer any questions. Okay, thank you.